dedicated to conveying value and interpreting great books worldwide. Hello, this is Sally's Book Club. Today we'll be exploring the book God's Scalpel. First, let's unpack the title. If God were to perform surgery on life, what would his scalpel be? Well, the most likely answer is gene editing technology. It's because even minuscule changes in genetic structure can dramatically alter life's characteristics. God's Scalpel chronicles humanity's journey to understand and reshape genes. The author, Wang Liming, born in 1983, is a biologist and professor at Zhejiang University's Life Sciences Institute. As a frontline researcher, he's also a masterful storyteller. Scientific research aims to solve problems like industrial pollution, cancer, and superbugs. Yet science often appears needlessly complex to the public. So Wang Liming seeks to reframe science's narrative, to tell its true story. He transforms scientific history into a riveting novel, empowering us all to grasp genetic truths at this pivotal moment when humanity rewrites life itself. Now we'll explore the book through 4 sections. Part 1. How Humans Uncovered Genetic Secrets. Part 2. Using Viruses as Genetic Scalpels. Part 3. Current Gene Editing Technologies. Part 4. The Future of Gene Editing. First, let's discuss how genes were discovered. Since ancient times, humans applied genetic principles by saving plumbed grains as next year's seeds, ensuring bountiful harvests. As the saying goes, plant melons, get melons. Plant beans, get beans. But why do offspring inherit parental traits? Greek philosophers like Democritus proposed pangenesis, pan meaning widespread, genesis meaning origin. They believed pangenes were invisible particles transmitted during mating, passing traits to offspring. When Darwin advanced evolution in the mid-1800s, he adopted this pangenesis concept. He theorized that parental pangenes fuse during reproduction, creating diverse hereditary traits. Clarifying traits, inherited biological characteristics, for example, double eyelids or single eyelids, straight hair or curly hair. Well, there's something puzzling about the theory of pangenesis. When parents have different traits, how do pangenes determine the offspring's traits? What color should the kittens be if a white cat and a black cat mate? Would they be gray or black and white, spotted? In the 1860s, Gregor Mendel, a pioneer in genetics, famously known as the pea fanatic, solved this problem. Mendel was originally a priest in Austria. He fenced off a plot in the monastery, planted peas day in and day out for years, cross-pollinated them, and recorded their traits. Eventually, he discovered that genetic information exists in discrete particulate forms that don't blend. The clearest evidence was that offspring of yellow and green peas were all yellow, but when those yellow peas were crossed among themselves, green peas reappeared in the next generation. Clearly, this shows that genetic information doesn't blend or disappear. It's just temporarily hidden. And when the opportunity arises, this hidden information stubbornly resurfaces. Unfortunately, Mendel's theory wasn't recognized at the time. It wasn't until the 1930s that someone linked his work with Darwin's theory of evolution and named Mendel's hereditary particles gene, which in Chinese is jian. This name was coined by Tan Jiazhen, one of China's first-generation geneticists. So now we know that genes are the material basis of heredity. The next question is, what exactly are genes? Where are they located? How can we find them? By the early 20th century, it was understood that organisms are made of cells, which contain macromolecules like proteins and DNA. Biological traits are expressed through proteins, whether pea color or cat color. It all ties back to proteins. Scientists thought genes must reside in some protein, but this guess was later proven wrong. This mistake was uncovered thanks to British pathologist Frederick Griffith, who conducted a serendipitous experiment in the 1920s. He was developing a vaccine for pneumonia. caused by Streptococcus pneumoniae. But there are two strains, smooth and rough. Only the smooth strain causes fatal pneumonia. Griffith injected mice with live rough bacteria and heat-killed smooth bacteria, hoping to trigger immunity without killing them. But both stimuli were too weak. The immune system ignored them. So Griffith threw caution to the wind and injected the mixed bacteria. This time the immune system reacted, but the mice soon died. Baffled, Griffith examined the dead mice and shockingly found live smooth bacteria inside. But he'd clearly heat-killed the smooth bacteria. Where did these come from? Griffith brilliantly deduced that genes from smooth bacteria had entered rough bacteria, producing smooth offspring. This accidental discovery accelerated the gene hunt. American scientist Avery refined Griffith's experiment. He removed proteins from smooth bacteria and mixed them with rough bacteria. Yet transformation still occurred, meaning genes weren't in proteins, so where were they? In 1952, two American scientists used bacteriophages, phage meaning to devour, bacteriophages. They labeled phage DNA and proteins differently, then let them replicate. Result, offspring inherited far more DNA than protein. This suggested genetic material was likely in DNA, but a key problem remained. DNA was known to be extremely simple, just 4 bases, so the question arose, how could complex genetic traits be transmitted by just 4 molecules? Since traits manifest through proteins, DNA had to connect to proteins. Proteins have 20 amino acids. How could 4 bases encode 20? Enter physicist George Gamow, a genius who proposed the Big Bang Theory, unexpectedly solving this biological puzzle. One day, he had an epiphany. If single bases couldn't handle 20 amino acids, what about combinations of multiple bases? 2 bases grouped together yield 16 combinations. 3 bases give 64, more than enough to handle 20 amino acids. Oh, well, inspired by Gamow, biologists had a sudden epiphany. Not only did they quickly prove DNA uses 3 base codons for amino acids, but they deciphered all 64 combinations. By then, scientists knew genes existed as base sequences in DNA, which directs protein production. Meanwhile, biologist Watson and Crick discovered DNA's physical structure, the now famous double helix. DNA resembles a twisting zipper, where teeth, bases on both strands pair perfectly. During replication, the zipper unzips into 2 strands. Each strand then builds a new partner following base pairing rules, creating 2 new double helices. That concludes the history of genetic discovery. To recapitulating key points, hereditary material is genes. Genes exist as base sequences in DNA, a double helical chain directing protein production. Now, part 2, how viruses act as genetic scalpels. Genes determine innate traits and impact health. Since genes direct protein production and proteins define bodily traits, faulty genes cause disease. Albinism exemplifies this. Patients lack a melanin-producing gene, causing total pigment loss. Thus, direct genetic surgery is the most effective disease treatment. Dr. Anderson at the U.S. National Institutes of Health pioneered this approach. His patient was a 4-year-old girl. Tor Ashanti had severe combined immunodeficiency, SCID, from a white blood cell gene mutation. SID patients lack immunity, even a sneeze could kill them. Back then, 2 treatments existed, injecting the missing protein, but this was temporary since proteins deplete, requiring repeated injections. The alternative was transplanting bone marrow, producing healthy white blood cells, but doctors couldn't find a match. Desperate, Ashanti's parents approached Dr. Anderson, embarking on an unprecedented medical adventure. Dr. Anderson's plan: extract blood from the girl, isolate white blood cells, insert healthy genes into them, then reinfuse them. Theoretically, this was sound. The challenge was delivering invisible genes into microscopic cells. And with billions of cells, you couldn't inject each one with microneedles. Dr. Anderson devised a risky solution. Using Maloney-Murine leukemia virus, he inserted healthy genes into the virus, using it to infect Ashanti's cells. Though deadly to mice, it's gentler on human cells. He also deleted most viral genes to prevent replication. After 12 days of treatment, the healthy genes in Ashanti started functioning. In 1990, humanity's first genetic surgery succeeded. With this precedent, later researchers naturally sought improvements. You might have realized Anderson's approach wasn't perfect. White blood cells aren't immortal, they gradually die. So when modified cells died, Ashanti needed repeat treatments. Trying to solve this. Let's rethink. White blood cells originate from bone marrow, specifically hematopoietic stem cells. If we modify these stem cells to continuously produce healthy white blood cells, wouldn't that be a permanent solution? In 1999, European doctors extracted hematopoietic stem cells from SCID children, modified them with viruses, and re-injected them. They hoped this would provide lifelong benefits without repeated treatments. In 2002, after nearly 3 years of follow-up, doctors finally confirmed that patients' immune functions had fully recovered. They confidently published a paper in academic journals stating that gene surgery targeting hematopoietic stem cells would greatly advance the treatment of rare genetic diseases. However, reality hit them hard. Just months later, a boy who received the treatment was diagnosed with leukemia. By fall 2003, 5 out of all treated children had been diagnosed with leukemia. As for the reason, it was actually quite simple. The tool doctors used for gene surgery was inherently a dangerous leukemia virus. Dr. Anderson, prioritizing safety, opted to modify short-lived white blood cells each time. Even if the virus altered genes unexpectedly, the cells wouldn't last long, minimizing harm. But successors targeted hematopoietic stem cells directly. These cells divide continuously, producing white blood cell offspring. If uncontrolled, it naturally leads to rampant proliferation. Science always advances through continuous failures. After recognizing the virus risks, people made improvements by using safer viruses and stricter clinical trials, taming this unruly scalpel. Proteins are made of amino acids, only 20 types. With just 4 bases, how do they correspond to 20 amino acids? The hero wasn't a biologist or chemist, but a physicist named Gamow, a genius who proposed the Big Bang Theory. One day, on a whim, he thought, since 4 bases alone can't handle 20 amino acids, why not combine multiple bases? 2 bases grouped yield 16 combinations, 3 gives 64, more than enough for 20 amino acids. Inspired by Gamow, biologists had a sudden epiphany. Not only did they quickly prove DNA uses 3 base codons for amino acids, but they deciphered all 64 combinations. By then, scientists knew genes exist as base sequences in DNA, which directs protein production. Meanwhile, biologists Watson and Crick discovered DNA's physical structure, the now famous double helix. DNA resembles a twisting zipper, where teeth bases on both strands pair perfectly. During replication, it unzips into 2 strands. Each strand then builds a new partner following base pairing rules, creating 2 new double helices. That concludes our journey through genetic discovery history. To recapitulating key points, hereditary material is genes. Genes exist as base sequences in DNA, a double helical chain directing protein production. Now, part 2, how viruses serve as genetic scalpels. Genes determine innate traits and impact health. Since genes direct protein production, and proteins define bodily traits, faulty genes cause disease. Albinism exemplifies this. Patients lack a melanin-producing gene, causing total pigment loss. Thus, direct genetic surgery becomes the most effective treatment approach. Dr. Anderson at the U.S. National Institutes of Health pioneered this concept. His patient was a 4-year-old girl. Ashanti had severe combined immunodeficiency (SCID) from a white blood cell gene mutation. Side patients lack immunity, even a sneeze could be lethal. 2 treatments existed: injecting the missing protein Temporary, since proteins deplete, requiring repeated injections, the alternative, bone marrow transplants, producing healthy white blood cells but no matching donors were found. That desperate, Ashanti's parents approached Dr. Anderson, beginning an unprecedented medical gamble. Dr. Anderson's plan: extract blood, isolate white blood cells, insert healthy genes, then reinfuse them into Ashanti. This plan is theoretically sound, but the challenge is delivering invisible, intangible genes into tiny cells. Plus, with billions of human cells, you can't inject each one with microneedles. So, Dr. Anderson devised a risky solution. Using Maloney marine leukemia virus, he inserted the normal gene into the viral genome, used it to infect Ashanti's white blood cells, and restored the gene in defective cells. This virus is deadly to mice but gentler on human cells, and he deleted most viral genes to prevent rampant replication. After the 12-day treatment, the normal genes in Ashanti started working. In 1990, humanity's first genetic surgery succeeded. With this success, later researchers naturally sought improvements. You might have realized Dr. Anderson's treatment wasn't perfect. Because white blood cells aren't immortal, they gradually die off. So when the modified cells died, Ashanti needed repeat treatments to re-engineer them. So how to solve this? First, let's rethink. White blood cells originate from bone marrow, specifically hematopoietic stem cells. So if we modify these stem cells to continuously produce normal white blood cells, wouldn't that solve it permanently? Innovative, clinical, European doctors extracted hematopoietic stem cells from children with combined immunodeficiency, modified them with viruses, and re-injected them. They hoped this would benefit patients for life, avoiding repeated treatments. In 2002, after nearly 3 years of follow-up, doctors confirmed patients' immune functions had fully recovered. They confidently published papers stating gene surgery on hematopoietic stem cells would advance rare genetic disease treatment. However, reality hit them hard. Just months later, a treated boy was diagnosed with leukemia. By fall 2003, 5 of all treated children had been diagnosed with leukemia. The reason was actually quite simple. The tool doctors used for gene surgery was inherently a dangerous leukemia virus. Dr. Anderson prioritized safety by modifying short-lived white blood cells. Even if the virus altered genes unexpectedly, the cell's limited lifespan minimized risks. But successors targeted hematopoietic stem cells directly. These constantly dividing cells, if uncontrolled, naturally cause rampant white blood cell proliferation. Science always advances through continuous failures. After recognizing viral risks, researchers improved this unruly scalpel through safer viruses and stricter clinical trials. Because it's incredibly simple, just design a CRISPR sequence and attach it to the Cas9 protein for precision cutting. As for CRISPR-Cas9 applications, they're virtually limitless. For example, Chinese and Korean researchers used it to remove muscle growth inhibiting genes in pigs, producing leaner pork. Researchers at West China Hospital modified lymphocytes to treat lung cancer. American scientists also used it to attempt excising HIV genes. As mentioned earlier, this technology truly ushered in the gene editing era. To summarize part 3, zinc finger patent barriers forced new solutions, leading to talon protein's emergence, which rendered zinc fingers obsolete, but CRISPR-Cas9 emerged victorious, currently humanity's most advanced gene editing technology. Gene editing development will never stagnate. Even today's advanced CRISPR-Cas9 has limitations. For example, the Cas9 scissors are oversized, larger than multiple zinc fingers combined. It sometimes mistargets and cuts similar gene sequences. We can't blame it. Designed to destroy invading phages, it naturally errs toward overkill. In short, gene editing technology still requires further scientific research. But we can confidently say that someday humanity will truly master God's scalpel. Finally, When the gene edit era truly arrives, are we prepared to bear its risks and impacts? In this book's conclusion, author Wang Liming discusses gene editing's future. It won't bring only benefits. Humans can treat diseases through gene editing and modify disease-causing genes to reduce risks. But extending this logic, could we modify genes to enhance our or our children's height, weight, appearance, or intelligence, or even create a perfect human? Not yet. Scientists aren't planning illegal activities. Remember Dolly, the cloned sheep born in 1996? Afterward, many countries banned human cloning, anticipating ethical dilemmas. Would clones be independent individuals? How would they face their genetic originator? Moreover, immature gene editing used recklessly for human creation could bring unknown catastrophic consequences. Of course, risks shouldn't halt editing development. Science ethics clashes are inevitable, and ethics often lag behind discoveries. For example, medieval Europeans terrified of plague believed viruses entered through bathing, making infrequent bathing a mainstream value. And Louis XIV, the Sun King, bathed only 7 times in his life. Anyone advocating bathing was considered morally bankrupt. So when science clashes with values, avoid snap judgments. Let's patiently await new discoveries. Well, that's God's scalpel. This concludes our book interpretation. Meeting notes are below the audio. See you tomorrow.